Sir, can we start the session, sir?
Yes, ma'am, you can.
Yes. Good afternoon to one and all present here in this forum for 1 week faculty development program on research prospects and progress in biological science. Learning gives creativity, creativity leads to thinking, thinking provides knowledge, knowledge makes you great. It's a famous quote by our beloved Abdul Kalam sir. With this positive wording, this is Priya, Assistant Professor, Department of Biotechnology, inviting you all for 1 week national level faculty development program on research prospects and progress in biological science. Now, may I call upon Dr. M. Kamraj sir, head in charge and convener of this program, to deliver a welcome speech. Sir, over to you.
Very good afternoon, honorable person in this virtual platform. I hope you all enjoyed the previous day sessions. On behalf of Department of Biotechnology, College of Science and Humanities, SRM Institute of Science and Technology, Ramapuram campus, it's a privilege to have eminent persons such as Dr. A.K. Sriklama with us today to deliver the talk on the plant productive biological investigation, the ways and means of biodiversity conservation. Ma'am, we really feel privileged to have you with us today. So I welcome you to this 1 week national level FTP program. Ma'am is Principal Scientist, Conservation Biology Division, Jawaharlal Nehru Tropical Botanical Garden Research Institute, Kerala. So she has ample research experience and in the medicinal plants. The further information will be shared in the introduction session. I'm here with officially welcome you the today's resource person, Dr. A.K. Sriklama, ma'am, once again. And we all are expecting to have a wonderful session from this lecture. Thank you, ma'am, for being with us today. Priya, ma'am, over to you.
Thank you, sir.
Shall I start the program? Are you audible? Audible?
Yeah, ma'am. Yeah, ma'am. Ma'am, 2 minutes, ma'am. It's an immense pleasure for me to introduce today's guest of honor, Dr. A.K. Srikala, ma'am, Principal Scientist, Conservation Biology Division, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Kerala. Our speaker has submitted a PhD thesis on pollen stigma interaction of some selected angiosperms under areas of specialization or plant reproductive biology. She started her career from the year of 1998 as scientific assistant. Until date, it is a successful one as a principal scientist. She is having research and development experience of over 26 years in both basic and applied sciences, covering all fields like plant reproductive biology, ecology, and taxonomy. She is engaging in analyzing various causal factors which leads to rarity in species, rescue and restoration of extinction-prone species in Western Ghats. She also assisted for establishing a field gene banking division and introduced these rare species as a part of development of package of practices for popularization and conservation. Recently, ma'am has completed one ecology project under the theme of plant-animal interaction, funded by Department of Biotechnology, Government of India. Our guest speaker has finished many successful projects and serving as a member for many professional organizations. Ma'am also guided 15 successful PhD scholars and made enormous publications in many reputed journals and also book chapters. Once again, I'm happy to welcome you, ma'am, for this 1 week faculty development program, and I hope participants will be enriched with the knowledge about plant reproductive biological investigations after your presentation. Welcome you, ma'am. Thanks a lot.
Thank you for the introduction.
Thank you, ma'am.
Okay. Shall I continue?
Yes, ma'am. Yes, ma'am. You can start.
Okay. Are you audible, my slides?
Yes, ma'am. Yes, ma'am.
Audible?
Yes, ma'am.
Okay. Good afternoon to everybody, respected faculty members, students. I am Dr. A.K. Srikala, Principal Scientist, Conservation Biology Division of Tropical Botanical Garden, Palod, Thiruvananthapuram, Kerala. The topic of my presentation is plant reproductive investigations, the ways and means of biodiversity conservation. India has been recognized as one of the 17 mega diversity centers of the world. Large number of plant and animal species occurring across the length and the breadth of the country is the evidence of its biological diversity. However, this plant wealth is eroding at a fast pace due to habitat degradation, overexploitation, climate change, reproductive constraints, etc. Reproductive inefficiency may also lead to the rarity of a species in certain cases. These combined effects over a long period accelerated the process of rarity and endangerment and finally led to reduction of resource base for sustainable utilization. What is reproductive biology? Reproductive biology is a branch of science which deals with the whole life cycle of the plant. It encompasses the study of pollination mechanisms, gene flow, genetic variation, and propagule dispersal between and within populations. Therefore, a detailed information on the reproductive biology of plants are essential for developing effective strategies for their conservation and sustainable utilization. Reproduction in flowering plants. We can classify the reproductive strategies into two: asexual reproduction and sexual reproduction. The mode of asexual reproduction is means of structures called vegetative propagules such as tenders, stolons, bulbs, tubers, etc. Sexual reproduction is an elaborate and a complex that shows that it is a slow process as compared to asexual reproduction. These are the modes of asexual reproduction: by stolons, by rhizome, by tubers, by bulbs, by suckers, etc. These are the best examples of asexual reproduction. Sexual reproduction in flowering plants, the reproductive role of flowers. Flower is the reproductive unit of angiosperms, which is meant for sexual reproduction. A typical flower has 4 essential whorls arranged on the thalamus or receptacle. These whorls are calyx, corolla, androecium, and gynoecium. Androecia and gynoecia are considered as reproductive structures. Androecia represents the male reproductive organ, and gynoecia represents the female reproductive organ. Androecium, the male reproductive organ of the flower. Each stamen consists of a filament and anther. Anther is usually bilobed, having 2 chambers called the pollen sacs. Pollen grains are released through the time of anthesis. Each pollen sac consists of 2 layers, such as pollen spore mother cell and tapetum. So the function of androecium is to produce pollen grains. Gynoecium is the female reproductive structure, consists of 1 or more carpels. Each carpel consists of an ovary, style, and stigma. Each ovule contains an egg cell inside an embryo sac. After fertilization, the ovule develops into seeds and ovary develops into fruits. This is the structure of an embryo sac. The top portion is the chelacelent. The bottom part is the micropylar end. 3 antipodal cells are seen. In the central portion, a central cell consists of 2 polar nuclei, 2 synergids, and an egg cell is present. The life cycle of angiosperm. A mature anther consists of lot of pollen grains. These pollen grains are ruptured at the time of anther dehiscence. They are carried away by wind and happen to reach on the receptive stigma. The pollen grains well adhered on the receptive surface, then grows through the stylar region, finally reach to the base of the ovary, enters through the micropylar region, and fuses with the egg to develop an embryo, and fuses with the central The second male gamete fuses with the central cell to form an endosperm. The endosperm provide nutrients to the developing embryo. Pollination. Pollination is an important phenomenon of plant reproduction. The transfer of pollen grains from anther to stigma of a pistil is termed as pollination. Successive pollination depends upon regular attractants of pollination. These are the major agents of pollination, that is wind, abiotic agents and biotic agents. Biotic agents are wind and water, and biotic agents are insects, birds, bats, snails, ants, and snakes. Strategy of animal-mediated pollination. Some plants support animal-mediated pollination. The flowers produced some visual cues such as colored perianth, stamens, colored staminodes, corona, etc. to attract the insects for pollination. They also produced some olfactory cues also. They produced some sweet scents. Pollen and nectar are the major rewards. Waxes, in addition to this, waxes and resins are also considered as rewards. Basic pollination syndrome. In some flowers have a particular color, odor, shape, and the system, the production of nectar that depends upon the pollinators. For example, in bat pollinated flowers are white colored. They open in the night time, produced a lot of copious amount of nectar, fragrance is there. Each plant has a special adaptation for pollination. Adaptations of animal-mediated or insect-mediated pollination. In such cases, petals are brightly colored with the guidelines. Scent is there, small amount of sticky pollen. They have a sticky stigma. Male and female organs are enclosed in the flower. You can see a beautiful flower, that is Impatiens coelotropis. We can see androecium, lower sepals, wing petals, etc. But wing petals act as a landing platform for insects. The second picture shows a spur. In the spur portion, the nectar secreting cells are present. At the time of anthesis, they produce a lot of copious amount of nectar in the spur portion for pollination. And the next one is insect-mediated pollination, endomophily. Bee pollination is also called melitophily. In such cases, flowers are showy, colorful, fragrant, nectar gates are present. In all the cases, and a landing platform is also present in the flower. Butterfly pollination is also called a psychophily. In such cases, flowers are showy, colorful, fragrant, nectar gates are absent. Moth pollination. Moth pollination, the adaptations of moth pollination, the flowers are large, white colored, fragrant, nectar gates are absent, usually tubular or spurred flowers. Fly pollination is also called mycophily. In such cases, this type of flowers emitted a bad smell. The color of the flower may be pale or dark brown in color. Nectar gates are not present. Flowers are funnel-shaped or complex traps. Adaptation of bird pollination. Most of the flowers are deep red in color. Most of them are tubular. They secrete a lot of nectar. Flowers vary in sugar composition. Adaptations of bat pollination. I explained earlier. The bat pollinated flowers are usually white in color, large. They usually bloom in the night time, produce copious nectar or pollen. Most of them are emitted fragrance. Adaptations of what are the adaptations of wind pollination? Wind pollinated flowers are small, numerous, perianth absent or non-showy. They produce a large quantity of pollen grains. Styles have branched for capturing pollen grains in air. Stigmas are large, feathery, and outside petals. Vallisneria is the best example of water pollination. After maturation, the male flowers are detached from the plant and float above the water surface. The stalk of the female flower is very long to project their stigmas above the water. In this way, pollination takes place. The difference between insect pollination and wind pollination. In the case of insect pollination, the flowers have large colored petals. They have a seeds, and they are seed-scented, have a nectary, have a sticky stigma. The female and male organs are enclosed in the flower. But in the case of wind-pollinated flowers are very small. They have no scent. They have no nectary. They have a feathery stigma to catch the pollen from the air. The female and the male organs are hanging outside the flower. Flexi-styly. Flexi-styly is an important phenomenon. This phenomenon was reported by a famous reproductive biologist, Dr. A.K. Kaul, in Picoraceae curva, belongs to the family Scorpulariaceae. The bilabiate flower of this species is heterostylus. The style is smaller than the stamens, therefore, flowers are cross-pollinated. Due to the absence of pollinators, the style elongates and just it touches the andrecium and traps sufficient pollen and ensures pollination. This is another example that is Passiflora edilis, our passion fruit. Due to the absence of pollinators, the plant itself has an adaptation for self-pollination. Its style elongates and just it touches the andrecium. In this way, pollination takes place. This is another example. This plant belongs to the family Malvaceae. Due to the absence of pollinators and harsh weather, its style curved and the stigmatic portion touches the andrecium. In this way, pollination takes place. There are two types of pollination: natural pollination and induced pollination. Induced pollination are carried out by artificial means. Natural pollination is two types: self-pollination and cross-pollination. What is self-pollination? The transfer of pollen grains within the same species. It is of two types, autogamy and geitonogamy. What is autogamy? Transfer of pollen grains from anther to the stigma of the same flower is called autogamy. For example, the members of crucifers. Geitonogamy, the pollination between different flowers of the same plant. For example, carica papaya, Musa indica, etc. Self-pollination is promoted by certain floral adaptations. First one is the homogamy, refers to the maturation of sex organs at the same time. Another one is the clustogamy. Clustogamy is a phenomenon in which the flowers do not open at the time of fertilization. Chasmogamy, flowers can remain closed, still self-pollination occurs. Once it over, they open. These are the major floral adaptations of self-pollinations. What are the advantages of self-pollination? Parental characters are preserved indefinitely. The flowers need not to be large. Fragrance and nectar production is not required. The major disadvantages are reduces variation in a population, results in the accumulation of deleterious alleles. This phenomenon is also known as inbreeding depression. Poor quality seeds will be produced. Cross-pollination. The transfer of pollen grains from anther to the flower to the stigma of another flower of different plants is called allogamy or cross-pollination. Pollination between flowers of different genetic constitution is known as xenogamy. This is the only type of pollination which during the pollination bring genetically different type of pollen grains to the stigma. For example, Impatiens Psilotrapis, Helianthus anus, etc. Major advantages of cross-pollination are produced new offsprings, promotes an increase in phenotypic variability within a population. Disadvantages, scarcity of pollinators may affect the sexual reproduction. It requires an agent for the transfer of pollen between individuals. Sequential events of pollen pistil interaction. The first one is the pollen arrival, then pollen adhered on the stigmatic surface, then goes to pollen hydration, pollen tube entry into the stigma, the pollen tube penetrate through the stylar region, pollen tube entry into the ovule and embryo sac. Finally, double fertilization takes place. Stigma receptivity. Stigma become receptive soon after anthesis, but the period of receptivity may vary from species to species. Stigma receptivity refers to the ability of the stigma to receive and support pollen germination. There are two types of stigmas, dry stigmas and wet stigmas. In wet stigmas secrete exudates, which contain certain extracellular compounds such as carbohydrates, proteins, phenols, amino acids, etc., which support pollen adhesion and pollen germination on the stigmatic surface. This slide shows some photographs of in vivo pollen germination. First one is the ultrastructure of the stigma. The second one is the pollen adhesion on the stigmatic surface. Third and the fourth ones are the pollen germination on the stigmatic surface. Fertilization, double fertilization. During fertilization, two sperm nuclei are discharged into the embryo sac, resulting double fertilization. The fertilized egg will develop into embryo. The fertilized central cell wall develop into endosperm. The ovary develops into fruits and the ovule develops into seeds. Seed dispersal. Seed dispersal is the movement or transport of seeds away from the parent plant is called seed dispersal. Seeds can be dispersed away from the parent plant individually or collectively, as well as dispersed in both space and time. What are the needs for dispersal? Minimize competition for light, water, etc., avoids overcrowding, colonizes new areas, increase chances of survival. What are the major agents of seed dispersal? Gravity, wind, water, and animals are the major agents of seed dispersal. Seed germination. Seed germination depends on both internal and external factors. External factors include water, temperature, oxygen, sometimes light or water. Internal factors include vitality, viability, and dormancy. The part of the plant that first emerges from the seed is termed a radicle, which allows the seedlings to become anchored in the ground and start absorbing water. There are two types of seed germination, that is hypogeal germination and Epigeal germination. In the case of castor and beans, the hypocotyl elongates and become curved. In the Epigeal germination, hypocotyl grows actively and become curved. Brings the plumule above the soil in hypogeal germination. In Epigeal germination, brings the seed above the soil. Cotyledons remain underground in the case of hypogeal germination. In the case of epigeal germination, after coming above the soil, the hypocotyl straightens. Reproductive biology of endemic and endangered species. I have studied the detailed reproductive biology of more than 50 impatient species. What is impatiens? Impatiens belongs to the family Balsamiaceae, comprising more than 1,000 species globally. In India, the genus is represented by 220 species distributed in 3 major centers of diversity, that is Western Himalayas, hills of northeastern states, and Western Ghats. 95 species available in peninsular India. More than 82 are endemic and confined to Western Ghats. I am going to show some endangered impatient species. The first one is beautiful flower impatiens viscosa. Impatiens platydina, it is a critical endangered impatiens, Impatiens cuspidata and Impatiens phoenicia. These 4 species were located in Munnar of Idukki district. Yellow colored flower, Impatiens darceli, which was located in Maharashtra. Impatiens fruticosa was located in Manjore of Ooty district, Ooty of Tamil Nadu. A beautiful flower, Impatiens silotropis. This species was located in Rajamale of Idukki district. Impatiens grandis, it was located in Kallar Valley of Idukki district. Pulcherrima was, Impatiens pulcherrima was located in Karnataka near Jog Falls. This species, another species, Impatiens trichocarpa. This species was located in Karnataka. Impatiens hensloviana was located in the Naimakad Gap of Idukki district. The phenology of Impatiens species. In all the cases, the species starts flowering in the month of July, extends up to December, and reaches a peak during September, October. These are the major pollinators of Impatiens species: Apis cerana, Apis dorsata, stingless bees, Trigona iridipenis, and different types of butterflies: Danus crisipus, Danus venusia, Thirumala limnaceae, Raptima baladus, Badamia exclamation, etc. All these species foraging in the daytime, but Macroglossum corythus, a hack monks foraging usually in the night time. Another floral visitor, Xylocarpa species, it collects nectar from the flower, make an injury on the spur portion, and collected nectar from that portion without facilitating pollination. These are the major pollinators of Impatiens grandis: Apis cerana, Apis iridipenis, dorsata, Epophila smaragdina, etc. This slide shows the major pollinators of Impatiens verticillata. These are the major floral visitors of Impatiens pulcherrima, Apis cerana, Apis dorsata, and some butterflies. These are the pollinators of Impatiens trichocarpa, platydina, and Impatiens silotropis. The fourth one is the Xylocarpa species. All these species are poorly distributed in the wild, mainly because of habitat degradation, climatic fluctuation, anthropogenic activities, and reproductive constraints. Reproductive constraints such as scarcity of pollinators, low insect visitation rate, inbreeding depression, poor fruit set, low percentage of seed germination, improper seed dispersal, etc. All these factors, either alone or in combination with others, are responsible for the narrow distribution of these species in the natural habitat. Okay. I have studied the detailed reproductive biology of some tree species also. This is Humboldtia decurrens, belongs to the family Fabaceae. The genus Humboldtia consists of 7 species and 2 varieties. This is a critical endangered tree species, Humboldtia unijuga. This is Humboldtia brunonis. It was located in Vaitiri of Wayanad and Tamarasheri of Calicut district. Another one is Humboldtia brunonis variety Rectapushpa. It was only located in Kakkayam. Another species, Syzygium maihendre, an endangered tree species, which was located in Ponmudi Hills of Agasthyamala Biosphere Reserve. Another species, Syzygium species. This is Eleocarpus tuberculatus, another endemic tree species located in Western Ghats, Garcinia imberti. It was located in Bonakad Hills of Thiruvananthapuram district. All these species are poorly distributed in the wild, mainly due to both biotic and abiotic factors. Reproductive inefficiency may be one of the reasons. The other reasons are also their habitat degradation, climatic factors, etc. Reproductive constraints such including low insect visitation rate, poor food set, low percentage of seed germination, poor natural regeneration capacity of the seeds, etc. Then moving to conservation. What is conservation? Conservation is the act of protecting Earth's natural resources for current and future generations. There are two types of conservation. Conservation of species in their natural habitat. That is in situ conservation. National parks, biosphere reserve, wildlife sanctuaries, sacred, those are the best examples of in situ conservation. Ex situ conservation, what is ex situ conservation? Conserving species out of their natural habitat is called ex situ conservation. Botanical gardens, seed bank, gene banks, pollen banks are the best examples of ex situ conservation. Biosphere reserves and national parks. First, the area that is suitable for the creation of a biosphere has to be identified and delimited. This requires surveys to collect data on key species. Property may have to be expropriated. A legal framework may need to be set up to control human activities in the area and in its immediate surroundings. The example of National Park, Irivikulam National Park, you can see the Nilagiri tar, an endangered animal conserved in the Irivikulam National Park. Another the plant is Promelanthus kundiana, belongs to the family Acanthaceae. This plant flowers once in 12 years. Advantages of in situ conservation. The species will continue to evolve in their environment. The species have more space, bigger breeding populations can be kept. The major disadvantage, it is difficulty to control illegal exploitation. For example, poaching. The environment may need restoring and alien species are difficulty to control. Botanical gardens are the best example of ex situ conservation of rare plants. I am working in the botanical garden. Botanical gardens are playing a key role as centers of conservation of plants. Botanical garden is a garden dedicated to the collection, cultivation, and display of a wide range of plants labeled with their botanical names. What are the objectives of a botanical garden? The botanical garden actually provides scientific research, horticulture, game blossom conservation, recreation and restoration, public education and popularization. The vision, major vision, conservation and sustainable utilization of the plant biodiversity of India, particularly of the Western Ghats, for the well-being of our people. Mission, to advance knowledge, enjoyment, and the conservation of plants through excellence in biodiversity research management, horticultural displays, and educational programs. This is our research institute, a tropical botanic garden, hold over 4,500 species, over 45,000 accessions. The bamboo citrus 69 species, cacti and succulents 250 species, ficus 75 species, medicinal plants 1,100 species, palmatum consists of 150 species, water plants 30 species, wild ornaments approximately about 120 species. This is our special. This is one of the specialty of our botanic garden. This is Victoria Amazonica, giant water lily. The lower surface of the leaf is very strong. This leaf can hold the weight of a baby. Paphiopedilla duri, it is also called lady slipper orchid. It was collected from Agasthyamala biosphere. This endangered plant was conserved in our garden. Our scientists developed a lot of saplings of this plant through micropropagation and conserved in our garden. Orchids. The photo of tiger orchid. We have the finest living collection of orchids in South Asia, comprising over 650 species and 150 hybrids. Wild species collected from Western and Eastern Ghats, the Himalayas, Northeast India, and Andaman Nicobar Islands. A dozen of new hybrids are also exhibited. This tiger orchid inflorescence consists of lot of flowers. This inflorescence can exist at least 2 to 3 to 4 months. Carnivorous plants, another speciality of our garden. Carnivorous plants which catch and digest insects are really wonders of nature. The Endemic hold 30 species of the attractive carnivorous plants. Cacti and succulents, 350 species of succulents, including around 100 species of cacti are grown in the cacti house and rock garden of my research institute. Bamboo setum, a beautiful bamboo setum harboring 954 accessions belonging to 69 species and one variety under 15 genera. The interesting ones are giant bamboos, the South American zigzag bamboo, climbing bamboos are there. Lesser known fruits. The research institute developed a very good conservatory of orchid, about 120 lesser known fruit plants in our garden. The first one is the in Malayalam Mutipalam, Bacoria cotalensis. Another one is the Syzygium palodensis. This species was newly discovered by our technical officer, Dr. E.S. Santosh Kumar. Seed bank is one of the example of ex situ conservation. Seeds can be maintained for decades or even centuries if the conditions are controlled. Not all species are suited to this treatment. Seeds needs to be regularly germinated to renew stock, or the seeds will eventually lose their viability. Duplicate stocks can be maintained. Seeds kept in seed banks do not evolve with the changes in the environment. In our research institute, we have a very good seed bank. Conclusion. The above mentioned information are very essential for the conservation and management of plant species and their dependent animal species in both in situ and ex situ approaches. Studies in reproductive mechanism will also help in developing strategies to preserve the genetic potential of rare plants, which are crucial for restoration and reintroduction. Thank you for hearing my presentation. Thank you very much, faculty members and my dear students. Thank you.
Thank you, ma'am, for your one.
Hello.
Hello. Thank you, ma'am, for your wonderful presentation
Okay.
on plant reproductive biology. Now the session is open for discussion. Participants, you can ask your doubts to the ma'am directly, or you can post the question in the chat box. Participants,
हम एक ही कमरे में सोते हैं ना। कौन सा तो
I think no doubts in ma'am's presentation. So without further delay,
Ma'am, excuse me, sorry to interrupt.
may I
There is some
request-
question, right?
Okay.
There is question in the chat box, ma'am. You can refer it.
Chat box. Okay. I will.
What are the major
ma'am,
threats?
what are the major threats? Yes, sir. Solunga, sir.
What are the major threats of venous condition?
Convulsia belongs to the family Fabaceae. One of my PhD students doing his PhD work, the reproductive biology of Cambodia brunonis. The major threat problems are fruit set problem. The flower fruit ratio is very low because only 5 to 10% fruit set was noticed in the natural habitat. I think the scarcity of pollinators, the low insect visitation rate, etc. Another thing is the some insect larvae and the monkeys destroyed the tender fruits in the natural habitat. The percentage of seed germination is also poor. Sometimes the fruit developed, but it contains without any seeds. That is the major problem. Some anthropogenic activities are also noticed in the natural habitat of Cambodia species. Okay. The plant is self-incompatible in our study. We conducted a lot of breeding experiments in the natural habitat. In our observation, the plant is completely partially self-incompatible. Only the breeding experiment showed that only a very low percentage of seed set was observed. Okay.
Thank you, ma'am.
Okay.
Any other questions, participants?
Students can ask questions.
Okay,
Ma'am,
ma'am. There
I
is no
think, no
further questions.
doubts.
You can, yeah,
Yeah.
you can end up.
Okay.
Is further delay, may I request Dr. G. Rajendran, sir, Assistant Professor, Department of Biotechnology, to give an vote of thanks.
Thank you, madam. Good afternoon, all. I am Dr. G. Rajendran, Assistant Professor in Department of Biotechnology. On behalf of the Department of Biotechnology, SRMIST, Ramapuram campus, I am glad to be standing here to express my vote of thanks. First of all, my most sincere thanks to God for making today's event a successful one. I wholeheartedly thanks to our today guest, Dr. A.K. Stigma, madam, Principal Scientist, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Thiruvananthapuram, Kerala, for accepting our invitation, sparing her valuable time and sharing her science and research knowledge in plant reproductive biological investigations, the way of means of biodiversity conservation. Madam has explained elaborately reproduction in flowering plants, mode of sexual and asexual reproduction, reproductive organs of plant, life cycle of angiosperms, strategy of animal pollination, difference between biotic and abiotic pollination, self-pollination, cross-pollination, fertilization, germination and its types, endemic and endangered plant reproduction, conservation and its types, biosphere reserve and animal and national parks, then botanic garden and its importance, seed banks and its uses. I hope all this information might be useful for all the participants to enhance your scientific knowledge and research. So once again, our sincere thanks to Dr. A.K. Stikala, madam, for her excellent lecture. Thank you, madam.
Thank you. Thank you very much. Faculty members and my dear students. Thank you. Thank you.
My special thanks to our management and our respected HOD for constant support and encourage to organizing this FDP program. I would also like to thank all my colleagues for the teamwork and the effort in this event. My special thanks to all the participants who made the event a memorable one. Your participation and discussion are always encourage us to organize event like this. Finally, I would like to thank each and every one of you for being here and making this day a wonderful lesson. See you again tomorrow in another session. Have a nice day. Thank you. Bye-bye.
Thank you. Thank you.
Thank you, ma'am.
Thank you. Thank you very much.
Thank you.
Thank you, ma'am.
Okay. Okay. Thank you. Bye bye.
Sir, post the feedback link.
Rajendran, sir.
Yes, sir. Yes, sir. Posting.
Participants are requested to fill the feedback form
Is available.
before leaving the session. Yes, sir. Yes.